Hi gang, Dr. West here. So this is the final lecture for this first exam, and we are going to begin to talk a little bit about neural development and how that is ultimately going to lead to behavior. So this should be really the last major piece of just sort of setting the stage. So up to this point, we've been doing a lot of just the pieces and parts of your nervous system. We've talked about the individual cells, both the neurons and the glial cells. We talked about how you have individualized sort of regions of your brain that are doing specific kinds of behaviors. We talked about how neurons communicate with electrochemical communication. So we talked about the electrical part, which was the action potential and all the details that went in with that. You just heard about the rest of the neurotransmitter part, which is the chemical part of that communication. And so in this last lecture here, we're going to take one more step back and we're going to look at sort of how the brain develops basically from the moment of conception on. And so what I'm actually going to do before I show you some more pre-recorded lecture from Dr. Bennett is 2 quick videos to sort of give you an overview of it. Again, that's just how my brain works, a little bit differently from hers, is I sort of like to see the big picture first. So one video, the very first one, is just a really quick sort of summary video. I like that one. It's very simplistic and it just kind of helps me get all the terms right in my head. But then the second one that I'm going to show you has a lot more detail, and you don't need nearly as much of the terminology and the detail. But what I really like about the second one is that you can see, again, the importance of the glial cells, right? Even from the very, very, very beginning, in helping the neurons to get themselves established, to get themselves even to the right location, as the brain itself is forming and becoming a brain instead of just random cells that have grown. So take a look at these first 2 videos, I said, and then we will jump over into Dr. Bennett's lecture, and we will be finished for this first unit. Thanks.
Welcome to 2 Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss early neural development. The development of the nervous system begins at around the 3rd week of embryonic development, when an area of the ectoderm, or the outer tissue layer of the embryo, thickens and forms what is known as the neural plate. This plate begins to fold inward, forming a groove called the neural groove. The sides of the neural groove, known as the neural folds, begin to come together. At the end of the 3rd week, the folds will begin to fuse together. By the end of the 4th week, they have completely fused together to form the neural tube, which will eventually become the brain and the spinal cord. As the neural tube closes, bulges and bends begin to appear and they gradually become more noticeable. During the 4th week, there are 3 of these bulges present. They are called the primary vesicles. They are the prosencephalon, which will eventually form the cerebrum, the mesencephalon, which will eventually become the midbrain, and the rhombencephalon, which will eventually become the rest of the brainstem and the cerebellum. The end of the neural tube will form the spinal cord. As the brain continues to develop, 2 of these vesicles further subdivide to form secondary vesicles. The prosencephalon forms the telencephalon and the diencephalon. The telencephalon will become the cerebral hemispheres. The diencephalon will eventually consist of the thalamus, hypothalamus, and other structures. The mesencephalon does not subdivide any further and will become the midbrain. The rhombencephalon will subdivide into the metencephalon and the myelencephalon. The metencephalon will become the pons and the cerebellum, and the myencephalon will become the medulla. The spinal cord will still form from the end of the neural tube. Next, the neural tube will continue to develop to look more like the brain. The telencephalon will grow more rapidly than other parts of the tube, and by 11 weeks, the brain will have a similar shape as to what it will at birth. Although it will continue to develop after birth, the brain at birth is structurally similar to a fully developed brain.
Shortly after fertilization, the fertilized egg or zygote begins dividing. By about 4 days after fertilization, a solid ball of cells called a morula forms. The cells continue to multiply, and by day 5, the cells separate into 2 groups, the inner cell mass, which will become the embryo, and an encompassing sac called the trophoblast, which will develop into the placenta. Trophoblast cells secrete fluid, creating a cavity, the blastocele, with the inner cell mass at one end. At this stage, the mammalian embryo is called a blastocyst. The cells of the inner cell mass eventually form two layers, the epiblast and the hypoblast. The epiblast is thought to contain the cells that will generate the actual embryo. Critical to the early development of all vertebrate embryos is the process of gastrulation, which occurs between days 13 and 19. Gastrulation results from the movement of cells toward the midline and then forward along the midline, forming a groove called the primitive streak. The end result is the production of the three primitive cell layers, the endoderm, the mesoderm, and the ectoderm. The nervous system develops from the outer layer, called the ectoderm. As this cell layer thickens, it becomes a distinct epithelium called the neural plate. Uneven rates of cell division form a groove called the neural groove that will become the midline of the embryo. Ridges of ectoderm continue to bulge on both sides of the midline. The tops of the neural ridges eventually come together, forming the neural tube. The interior of the neural tube becomes the fluid-filled cerebral ventricles of the brain, the central canal of the spinal cord, and the passages that connect them. By about 24 days, the three major subdivisions of the brain are discernible. These include the prosencephalon or forebrain, including the telencephalon and diencephalon, the mesencephalon or midbrain, and the rhombencephalon or hindbrain. The progenitor cells of the neural tube are known as neural precursor cells or neural stem cells. This represents the first stage of neural development, neurogenesis, when undifferentiated cells undergo mitotic divisions to produce either new stem cells or neuroblasts that will eventually differentiate into neurons. The cells undergo a stereotyped pattern of cell movements as they progress through the mitotic cycle. Eventually, the dividing precursor cells form a closely packed layer of cells called the ventricular zone. Eventually, some cells leave the ventricular zone, forming a second layer of cells called the marginal zone. Later, an intermediate layer develops as the wall thickens. Cells in the intermediate zone will then begin to differentiate into both neurons and glia. As the number of cells increases during the period of neurogenesis, the newly formed cells must move even greater distances. The massive movement of nerve cells to establish distinctive cell populations represents the second stage of neural development, cell migration. Cells do not move in an aimless, haphazard manner. Cells in the developing cortex move along the surface of a particular type of glial cell called radioglia. Like spokes of a wheel, these radioglia cells extend from the inner to the outer surfaces of the emerging nervous system. The radioglial cells act as guide wires, and the newly formed cells creep along their processes. Once cells reach their appropriate destination, the third stage of neural development begins, differentiation. At this point, the cells begin to use or express particular genes. This allows the cell to acquire the distinctive appearance and functions of neurons characteristic of that particular region. As the process of differentiation proceeds, the fourth stage of neural development begins, process outgrowth. The biggest change in brain cells early in life is the extensive growth of axons and dendrites and the proliferation of synapses. The neural environment also greatly influences nerve cell differentiation. In vertebrates, young neural cells seem to have the capacity to become many varieties of neurons, and the particular type of neuron that a cell becomes depends on where it happens to be and what its neighboring cells are.
Lecture for exam 1. This is a review of the neurodevelopment and how it's linked to behavior. Important things to understand about some fundamentals with regards to behavior and brain structure is that we understand that if the structure is not developed, then the behavior can't occur. So when we see a behavior develop, we believe that what's happening is the structure of the brain is being fine-tuned, or the underlying connections are being laid, basically. So we do this by looking at studies where you look at brain development, you can scan it and those types of things, and then you can predict social development. And we can see that when adolescence age. We can also see that in adolescence and toddlerhood, where there are changes in behavior that there are also then changes in the structure of the brain. And I'll have a video that I will post that you guys can look at. The other pieces that we look at is that when things that are behavioral, like language, when that develops, the idea is that the brain is then in that area developing and strengthening, and the underlying circuitry is emerging. So finally, what are those factors that play into whether the skill develops or the underlying structure develops, or whether the underlying develop is laid to allow the behavior to be created? A lot of things go in. Aging, hormones, genes, experience, injury, all those types of things have a major impact. So what we'll talk about now is just kind of some normal natural development. So what's interesting is when we look at the in utero environment for many species across them, and this is just an example from the textbook, where you have at some point in an early zygote stage, the salamander, the chick, and the human all look really similar. I know this is a drawing, but this is an accurate representation of different stages of development. And this is to support the theory of evolution. We talked about that in the first chapter, that the evidence appears that that we have evolved from a common ancestor all the way from a single-celled structure. And this type of development that we see in utero also supports that, because we go through stages. At some point in human development, we have gills. We obviously lose them because they're no longer effective because we don't live in water, but those are the types of things that we see. So looking at a human development, the zygote is that early first period where it's just been fertilized to 2 weeks, then it goes into embryo, which is 2 weeks to 9 to 8 weeks, and then at 9 weeks, which is about the end of the last trimester, not completely, I guess it's about 2/3 of the way through the first trimester, then it becomes a fetus. At week 3, after conception, so just in that early embryo stage, we start to see the neural plate forming, and that's what gives rise to the nervous system. Creates the neural tube, and so on and so forth. So here's an example. This is 28 days after conception or fertilization. You can see that the underlying structures are starting to develop with regards to the major structures of the nervous system. So you got your forebrain, midbrain, hindbrain, and then the spinal cord. And this is all happening, you can see, really early. If you think about females who may or may not be trying to get pregnant, they may or may not have as much of a understanding about where their body is in their cycle. To 25 days, a female may not even recognize she's pregnant, and look, the nervous system's already starting to, the groundwork's being laid. But you can just see it's a process that occurs, and it matures over time. At 9 months, all the structures are in the right places, but they're not fine-tuned. What we know about development is that you have to have experiences for the nervous system to be trained. And if you think about the in utero environment, there's not a lot of sensory perception going on, right? Because it's in a dark, warm, wet environment. Not a lot of sensations. Think about it, if you get in a bathtub or you swim in a warm pool and you put yourself underneath it. You hear things, but they're all muted. You really can't see things when your eyes are closed. And if it's at night and you're in a swimming pool with no lights, right? Think about that environment. There's really no sensory input, very little sensory input anyways. One of the things you'll see is that as the nervous system develops, there's the neural stem cells, which are just like any other stem cell in the body, has the potential of becoming any neuron, any glial cell. They're highly active in utero, and they're responsible for basically creating all the different types of cells. And we'll look at pictures and talk through this a little bit more. You've got your stem cell. It has the ability to go off and create more of itself, but it also becomes a progenitor, which means it's the beginning of differentiation. There are 2 different types of cells it can differentiate into. There's the neural cells, which go on and create neurons, or the glial cells, glioblasts, which go on and then create the support cells, the oligodendrocytes, the astrocytes, the microglia, those types of things. How do they know what to become? Well, this is where we're going to touch into epigenetics. Just a little quick touch on it. Epigenetics is a phenomenon that occurs in natural development of a species. In utero, for humans, it happens. It's how a heart cell knows to become a heart cell instead of a skin cell. It's how the brain, the nervous system, the neuroblasts know how to become that instead of becoming your bone osteoblast. It's basically what happens is all of our genes, all of our DNA, and each one of our cells, the only cells that don't have it are red blood cells, but every other cell in our body has DNA, and it all has the same exact DNA in it, at least roughly, 99.9%. And what ends up happening is, depending on where these methyl groups are, and this is just a picture showing you, giving you an idea, if there is a methyl group on, it will alter whether or not that gene is expressed. And there are chemicals that get exposed to the cells that will change the methylation and will change the future of that cell. But we also know that there are things like stress. So the mother's stress, she's under the stress hormones, the cortisol, those types of things can be influenced to what happens at the fetus level and how the cells end up developing and changing its course, which is something interesting to think about. It's those different factors that the cells are exposed to that drive whether or not it becomes whatever cell. So for the nervous system, there are different compounds that can support growth and differentiation. And here are just two examples so that you have an idea of the different types of things you can see. So there are epidermal growth factors, which takes a stem cell and pushes it into a progenitor state. And then there are, if it's exposed to basic fibroblast growth factor, the progenitor cell, it will then become a neuroblast. If it's exposed to something else, it'll go on and develop whatever it's been directed to. So a lot of the development of a human being requires on the certain chemicals being expressed at the right time. It's actually quite amazing. If you really want to have your mind blown, is to really get into some developmental biology, because you're going to learn that it's a miracle that we as a species continue to survive because of all the things that have to happen at the right time for a healthy, functioning human being to be born. It's pretty amazing. All right, so here's just a quick rundown of all the stages of brain development. The neurogenesis and gliogenesis, that's the creation of cells. This is very, very active during in utero experience. Neurogenesis and gliogenesis, neurogenesis primarily, we believe continues for life, although it is greatly slowed down after birth. Cell migration is responsible for helping their radioglia, and this is really the only time we see radioglia. Their sole responsibility, they're a support cell. They will help guide neurons to get to where they belong, and this is going on in the first trimester primarily. So here's a picture that will show you roughly what's happening. So you've got this cross-section of the brain, and it's showing you how these radioglia, which are represented by these blue, almost tube-like cells, they help guide the cells that are being created down here in the subventricular zone and migrate them out into the cortex, where they will go on and develop the ability to become neurons and start connecting and creating circuitry. Differentiation, we believe, is primarily complete by birth. So those first three steps of brain development are primarily found during the in utero phase. After differentiation, then you have maturation occur, which is where the dendrites and the axons grow. You have synaptogenesis. That is where the synapses are starting to be created. That's where the dendrites and the axons are starting to connect. And then there's a phase where the nervous system actually starts to fine-tune itself, and there's cell death of neurons that aren't necessary. There's the removal of synapses that aren't functional, that aren't helping that being survive. And then there's myelinogenesis, which is the formation of myelin. So that finishes out the development of the nervous system. And then we move, and I'm going to show you a graph here that will show you where the stages 4 through 7 occur, with regards to the development in utero. So 1 through 3 definitely happen in utero. Primarily, those are all done by the time the fetus is born. Now, cell maturation, migration, differentiation, those are the next steps. And what we have here is this graph's kind of showing you have conception, so this is fertilization, and then birth is all the way over here. You'll notice that neurogenesis is highly active early on, and then you have this migration and differentiation occurring and the neuronal maturation. So this is where the cells are becoming ready to be activated and ready to engage the fetus once it's born, and as an infant, its ability to engage with the environment. That's laying the groundwork and getting that set up. This is just showing you that roughly as the body weight increases, the brain weight increases. It seems to be pretty proportional. Now, neuronal maturation, this is where you start to have dendritic growth and axonal growth, where neurons are starting to communicate, trying to get connected with each other and trying to make connections and fire. So if you've never seen it, it is fetus, while they are in utero, they will move around. So they have the ability to have motor control. not very well directed, as you may have ever experienced, an infant, newborn. There's no head control. There's not very strong muscles, all those types of things, because they haven't been used, right? They've been in an environment that they've been buoyant. So they haven't had to fight gravity. They haven't had to do all the things that once they're out of the womb, they have to. So dendritic growth is often very slow. Axons grow much more quickly. So synaptic development, this is something that goes on forever. It starts off really in maturation, so later stages of the in utero environment, but continues to go on after birth. And it's actually one of the biggest things, a lot of the training that goes on in that first year, where the infant comes out pretty pathetic, not able to do anything, not able to control anything, to a 1-year-old who can often say a few words, has the ability to walk or at least crawl, definitely sit up. So all of these types of things are developing over that first year, and it's pretty dramatic to see the changes that occur. And one of the first areas that become well connected and integrated is actually the visual cortex. And this is, when you think about the brain and you think about the way in which it develops and becomes synaptically complex, it goes from the back to the front. So the occipital is the first, and then you kind of move your way through the parietal temporal, and then the frontal lobe, and the prefrontal is that last part to really become well connected. And this is something that we see in adolescence, and we'll talk about that in the video. So cell death and synaptic pruning. This is something that goes on as soon as the nervous system starts interacting with the world, the outside world, and it really causes the brain to become more efficient. At least a well-functioning brain will start to prune and get rid of connections that are no longer effective. One of the things that happens during development is there's a lot of redundancy created, so that if one neuron dies for whatever reason, there's another one behind it to help create the next connection. But part of that, part of infancy into toddler and all that social and motor and thinking development that occurs in those stages is the simplification actually of the nervous system, in a way, because there's way more there than it needs, and it starts to remove connections or remove neurons that don't end up getting connected. One of the processes that we talk about is it's pretty much a hypothesis. We see it in some animal models, but we've never really seen it in human, because for good reason we can't do this type of work, because you usually have to kill the animal to look at its brain post-mortem. And there's this theory that there's neural Darwinism, based off the same philosophies of survival of the fittest in animal species. And this is basically that a neuron that is sending the message at the right time to create reinforcement is the one that's going to receive the growth factor. When it's activated, the growth factor is going to be released, and it's going to maintain that neuron and strengthen its synapses at the dendritic end and also the axonal end. And that neuron will survive, and the ones around it that aren't well connected are the ones that will be removed and go through apoptosis, which is programmed cell death. All right, moving into glial development. This is where you have the astrocytes and the oligodendrocytes develop and are created. Now, one of the things that we know about the development of the brain is that astrocytes are developed early on and help provide the structure. Oligodendrocytes are also created, but their myelination isn't as well established. In utero, it does occur, but it is something that happens a little bit later on. So oligodendrocytes form the myelin, and the myelin is really important in speeding up and connecting and maturing the cerebral cortex and its functioning. It is also responsible for driving simplification of the brain and increasing the circuitry and increasing the speed at which things function. We see this going on throughout development, right? So when we're talking all the way from infancy through toddler, through childhood, into early, late childhood, so elementary school, and then you get into junior high or middle school, and then junior high and senior high, where you've got the adolescent stage. That is where we have the kind of last major maturation of the brain occurring. And many of you, if you're a traditional college student, your brain is actually still creating and refining its functionality. And this seems to slow down or finish out usually about 25. Is that the final connections, especially in that prefrontal cortex area, finally, if they're going to become established or be created, they're going to do it by roughly the mid-20s. So here's an example, and you can see how this shift occurs. So if you recall, the gray matter is the cell bodies and the dendrites, and the white matter is the axons, because that's where the myelination occurs. And what you'll notice, these lines are showing you as individuals age from about 7 to 19. So we're talking about basically the early peri-adolescence, before adolescence, prepubescence, through puberty, and then into your early 20s. The pink line happens to be the male line, and the blue line is the female line. They both follow the same trajectory, where there's an actual loss of volume overall, because, as I mentioned, the brain is getting more efficient and it is getting rid of things that it's not using, that are redundant or that are not beneficial. And what you'll notice is that as you hit adolescence and puberty, you really, the volume of gray matter starts to go down, but you're not losing a dramatic amount of volume because your white matter is increasing. Now there's a video that you need to go watch. It's not on the H drive. My apologies on that, but it is posted beneath this video for you guys to watch it. It's very interesting. It's a woman who does this research and talks about how we're able to capture the transitions that we see going on in the brain nowadays with fMRI. As I mentioned, infancy, the ability to, if you've ever been around an infant and toddler, you can see when they are able to finally start linking things and how much better their life gets. And how much better the parents' lives get, right? So if you are a supporter of the binky or the pacifier, for the first time that infant is able to get a hold of that binky pacifier and put it in if mouth and soothe itself, it's an amazing moment. Not only does the infant have a positive reward that is reinforcing that behavior, but it also helps the parents a lot. If you've not been one, you will understand this in a few years when you do choose to have children. Then if you keep moving on, the reinforcing behaviors of being able to grab the toys they want or grab the food they want. Again, there are messages, there are circuitry that once you're successful at getting that pincer together, right, that finger and thumb together on that Cheerio, once you're able to do that and then get it to your mouth, oh my gosh, the flood of dopamine in the brain is amazing, and it's going to reinforce whatever circuitry was just fired, so that the behavior can be repeated over and over again until, as an adult now, you can get food in your mouth pretty regularly without any problems. So again, here's just kind of talking through what we just discussed about how the circuitry is reinforced, and that is done by myelination. Once that myelination occurs, that is going to be the fast loop, and it's going to just now become part of that wiring, and it's going to connect the motor to the somatosensory area, which is also going to connect back to the motor area. So there's just all this going on and really kind of strengthening. And as we talk about in section 2, those different sections, we'll understand about the motor cortex, how the thickness and how that changes over time. Another thing that happens early on that we can use as an example of this neuronal development and really gaining understanding is language. Usually the onset is a few words, maybe before one and two, huge explosion of learning of words and being able to communicate them. One of the things we're starting to realize, and it's through research that's showing, is that children are actually understanding and hearing language a lot earlier than they're actually able to create a response. So when you think about that, right, and we'll talk about hearing, part of language is listening, and the other part about language is being able to communicate back. And what we think is happening is that infants and toddlers are really able to hear and understand the words that are going on around us, but they're unable to have the motor control to create the words to speak back. And that's what just develops a little bit later. So you need to be wary about what you're saying around an infant, much younger than you probably think you need to. The other thing we know is that the language areas of our brain are pretty well established by age 12. Now, if you think back to your training, if you're a typical U.S. student, when did you start a foreign language if English is your native language? You probably started at middle school. How old are you in middle school? Yep, you are 11, 12, 13, right? We introduce in our education system a foreign language at the time in which the brain is actually almost done with its elasticity in this area. It's not completely, because clearly people learn languages after age 12, but it becomes immensely more difficult. So if we take the research we know and apply it to the education system, we would actually be teaching that foreign language or a second or third language early on in development. So those of you who happen to have grown up in a bilingual home or trilingual home, you are at a much better place to be able to learn a fourth and a fifth language than someone who has only been exposed to English up through ages 11 or 12. The area of your language, your temporal lobe and the acquisition of that area is much more plastic and has a lot of ability to adapt than somebody who's only learned one language. So going to some developmental stuff, Piaget has his stages of cognitive development, and what we actually find is that what he observed and what he was able to identify as major markers in development of sensorimotory, of thinking, concrete thinking, those types of things, actually map onto physiological maturation of the brain, which is kind of cool to think about it. Like he was able to observe it and describe it, and now we're actually able to find the physiological understanding of the changes in the brain that are supporting the types of cognitive development that he was talking about. Just neat factors. Now, there are critical periods that are, where we haven't pinpointed them at 100%, but there are periods where expansive work is going on in the brain and setting up and laying down trajectories. And we only really get to see the repercussions of this development not going correctly when there are severe traumatic experiences or abuse that occurs. So an example is there are children who have been put in closets and raised without any type of sensory experience. And when they're found or when that situation's corrected, there's only a certain amount of development that they can actually go through. And we see that. We see it in animal models. We can create it there. Here's an example of a study that was done that may or may not have been done recently. Basically what they did was they covered one eye or kept one eye of a kitten's shut. And it turns out when you look at the abnormal side, it's the left eye was sewn shut, which means the right side of the brain, when we get there, we're going to talk about contralateral stuff, didn't get to develop the branching that was necessary to have the complexity there for that portion of the brain to develop. And even when you open its eye in adulthood, that area is unable to overcome that lack of branching. It doesn't do it very efficiently. There's probably some growth that goes on, but not a lot. So we talked about sensory deprivation, where you're removed from light, you're removed from sound. There's also deprivation of early experiences, which if you're locked in a closet, you're also not getting social experience. Therefore, is it a sensory lack of light and sound and interaction? Is that all due to also having a lack of social and being able to separate the two? It's very difficult to differentiate those. However, there are individuals who, and the examples are typically, there's been some orphanages where children have exposure to light, have exposure to other kids, but they're not touched, they're not hugged, they're not loved, they don't have that social connection. And that can have major impacts on the intellectual and social behaviors of that child. We can create this in animal models, and here's an example of one of these things that we can see. And in rodents, the rodents, if you were not aware of this, are highly social animals, both rats and mice. Rats are a little bit more social than mice are, but anyways, they are communal animals. They like to be with other of their kind. But one of the things that happens with research is they're often housed separately in their own cage. Individuals who were interested in studying what is this early life period, is it important for enriching the brain? And what they found was that A is an example here in this photo of an enriched or complex housing environment where you have novel toys to play with, you have other rodents to interact with, you can get exercise, you can climb, you can do all of these kinds of things. And this complex environment actually creates a more well-integrated neuron with more complex dendrites and axon connections and those types of things in the brain, where a rodent was housed by itself with no access to exercise or maybe very limited access to exercise on its own solo, has less complex brain development. It's very intriguing, right? Another example are animals learning their behaviors with regards to how do they set up their home. So this is an example of a rodent who grew up in a house, a group environment, and it was taught and learned how to create a nest. Now this white puffy stuff here, I'm going to show you in a moment what the rodent was actually given. And this was a group housed rodent, and it knows and it's trained and it gets the ability, it knows it needs to create this warm, fluffy environment if it's going to procreate in those types of. Now this rodent, unfortunately, was individually housed. And if you see this white gauze-like looking material, that's what this was. So the rodent didn't know what to do with it. So it just stood on top of it. And it's nesting, right? So it didn't learn, it didn't pick up the behaviors it needs to be successful to then have its next generation. So this is what we're talking about with neuronal development and the sensory deprivation or social deprivation. It leads to these abnormal, quote unquote, abnormal behaviors, which it clearly looks like for this rodent. It's not going to have a good nest for it to have its young in and those types of things. There are 2 videos that you should watch. They cover different experiences that humans have gone through, get at this abnormal development that can occur due to sensory and social deprivation. All right, that's the end of the lecture. Hope you enjoyed it. And this is the end of the material for exam 1.
Officials in the Los Angeles suburb of Arcadia have taken custody of a 13-year-old girl, and they say was kept in such isolation by her parents that she never even learned to talk. The girl still wore diapers and was uttering infantile noises. A social worker discovered the case 2 weeks ago, but the authorities are hoping she still may have a normal learning capacity.
Among the first to see the child was Temple City Detective Sergeant Frank Lindley.
I already knew that the child was 13 and a half years old. And I took one look at her, and she wasn't much bigger than my daughter Beverly, who had just turned 7 about 3 months earlier. And I really had a hard time conceiving of the idea that the child was the age that she was. The child obviously had been severely mistreated. She was still in diapers, couldn't walk. She had no verbal skills at all at that point. The last time I was on this street was probably 30 years ago. Yep, there it is. Hasn't changed much. The backyard looks the same. It's all weeds and dead grass. Looks the same as it did in 1970.
The house belonged to Clark Wiley. A loner, Clark had turned his back on the world after his mother had been killed in a hit-and-run accident. After the accident, things in the Wiley house would never be the same again.
The house was completely dark. All the blinds were drawn. And there were no toys, no clothes, nothing that would ever indicate to you that a child of any age lived there. The child's bedroom was back in this corner. That was the bedroom. The windows were covered to about 3 inches from the top, which were the only natural light that had ever come in there, and all the time the child was in the bedroom. The entire furnishings of the bedroom consisted of a cage with a pull-down chicken wire lid and some type of piece of wire securing it when they closed it down. There was a potty chair with some kind of homemade strapping device.
For 13 years, Jeannie had spent her nights locked in bed, her days strapped to a potty chair. During that time, Clark had ordered his son John and wife Irene never to talk to her. In her darkened room, she had led a life of near total isolation. Even close neighbors were completely unaware of her presence.
We came home from work and the police was here and they came to question us. That's when we found out, what happened and, that they had a little girl. Nobody knew before. And when we found out what happened and how she was treated, everybody was shocked and just unbelievable.
For their whole marriage, Clark had imposed his will on Irene. And blind with cataracts, she had been too scared to resist. But one day, something broke. While Clark was out buying groceries, she seized her chance and fled. It was the first glimpse the world would have of Clark and Irene's dark secret.
I met Clark and Irene at Temple City Sheriff's Station. They were both under arrest at the time. When we interviewed Irene, she would make no mention of the family whatsoever, particularly the children. I attempted, along with my partner, to interview Clark. He refused to talk to us. He wouldn't say a word. He never even acknowledged that he understood what we were talking about.
Mr. Wiley?
Yes.
Why did you keep your daughter in a room?
Mr. Wiley has no comment.
No comment.
We haven't had time to discuss the charges. We haven't even seen them.
Unable to face the truth, Clark took matters into his own hands.
This morning, the authorities reported that 70-year-old Clark Wiley shot and killed himself just before he was to go to court and be arraigned for child abuse.
After 13 years, Jeannie was at last free. And for scientists, she was just the case they had been waiting for. For 13 years, Jeannie had lived a life of complete isolation. Raised in a city bedroom, Jeannie was as much a feral child as if she had been brought up by wolves. At 13, she was the size of a 6-year-old. Worst of all, she had never been taught to speak. The question now, could she ever learn? Jeannie's case was so scientifically important that the government funded a team of scientists to help answer the many questions she posed. Two of the scientists who would become especially important to Jeannie were child psychologist James Kent and linguist Susan Curtis. Neither had ever encountered a case as extreme as Jeannie's.
We looked at her as a newborn in a way, even though we know she hadn't. She came with 13 years of memories and experiences, not all of them wonderful, most of them not, I think. And so we thought we needed to start to expose her to what the world was going to be like for her outside the hospital bed.
To Jeannie, everything was a new experience.
We did what you would do with your own kids if you were introducing them to the world. You'd take them out and hold them up and show them, and sort of judge from how they reacted to whether this was too much or not enough, and you could move on and do the next thing.
Jeannie was making amazing progress. As the experts looked on, they realized that she might be the answer to the question that had troubled science for so long.
So we seized this wonderful opportunity that she provided us in as loving a way as we could, but using it to finally get our chance to address head-on specific hypotheses and notions about human language and the human mind.
These hypotheses were based on the latest ideas about how children's brains developed. According to the theory, young children could only learn certain things at certain times, called critical periods. Language was one of these critical periods, and according to the theory, Jeannie, who was now a teenager, had missed her chance forever. But incredibly, Jeannie seemed to be proving the theory wrong. As this footage shows, Jeannie was blossoming. Not only was she delighted by the world around her, but she was learning the words for the new things she was seeing.
She was extremely interested in everything around her. She wanted to know the word for everything around her. She wanted to engage people all around her. She was not mentally deficient. Her lights were on, and everyone who worked with her, from teachers to therapists to me, knew that she was not retarded. It was clear as day. And as she began to learn more and more words, hundreds of words, much more rapidly than I ever imagined, and stringing them together, I began to think maybe I will be wrong. Maybe she will be the one that will prove that this hypothesis is incorrect.
But Jeannie could not escape the effects of her past so easily. She was still haunted by her traumatic upbringing, trapped by the memories of the awful fate she had suffered. And linguistically, she had stopped making progress.
She learned tons of words. She has an enormous vocabulary. But language is not words. Language is grammar. Language is sentences. How do you make a sentence? What can be a sentence? What is a sentence? How do you automatically know something's a sentence? So it wasn't because she was cognitively deficient. In other respects, it was because she was cognitively deficient in this island of human mind, the mental faculty that we call grammar.
At the time Jeannie was found, brain science was in its infancy. But today, we have a much clearer picture of what actually happens in cases of extreme neglect like Jeannie's.
In Jeannie's brain, the left part of her brain, her cortex, that has those neural systems responsible for speech and language, because she never heard any words and because she was never spoken to very often, they didn't get stimulated. And because they weren't stimulated, they got smaller and less functional and disconnected, and ultimately that part of the brain literally physically changes.
Today, with modern imaging technology, we can actually see what happens in the brains of feral children, and the effects are shocking. Without normal stimulation, their brains are smaller and malformed, and the earlier this neglect begins and the longer it carries on, the worse the damage will be. Starved of stimulation, Jeannie's brain had simply not developed the capacity for language. And now that she was a teenager, she would never be able to learn. Despite this, Jeannie continued to be a close part of everyone's life. But there was more trouble ahead.
Children have to belong to somebody when they grow up, and she was still a child, and she needed a family to belong to. So that's what we would have liked, a family that she could belong to. And that's not what happened, unfortunately. What did happen is about the worst outcome I think we would have envisioned.
On her 18th birthday, Jeannie moved back with her mother, Irene, into the house in which she had been so terribly abused. But after only a few weeks, it was clear that Irene couldn't cope. From here, Jeannie was moved into state care with terrible consequences.
I was a student, and people wouldn't listen to me. People who needed to intervene did not listen to me. And so I spent lots and lots of time on the phone pleading with people to intervene and save this person who had had the worst experience of deprivation and isolation in all recorded medical history.
Jeannie moved from home to home, sometimes with the very people who served as her therapists. This potential conflict of interests raised tensions among the many people involved in her life, and a tug of war erupted over the child. As Jeannie's condition deteriorated, Irene decided that Susan Curtis and the other academics had become too close to Jeannie. A lawsuit followed.
I went from being asked to be her guardian to 1 week later being prevented from seeing her or phoning her. And ever since then, I've been prevented from having any contact at all. So although I have lots of, I'm still a scientist, I'm still interested in knowing things about her language now and all kinds of interesting things I would like to pursue academically. Primarily, I would just like to see her.
Now, a ward of the court, Jeannie lives in an adult care home somewhere in Los Angeles, prevented from seeing the people who once meant so much to her.